Right, good afternoon everyone and welcome again to the weekly COVID webinar hosted by the Department of Medicine with the assistance of Project ECHO, University of New Mexico, extending their license to us on behalf of Wendy Spearman and myself, Mark Sundrup. Good afternoon to everyone. Full program this afternoon. Again, our thanks to all the work to make this possible. Cheryl Valentine from Project ECHO, Gastro Foundation, Karen Fenton, as well as Sahana Rangel and Claire Jeffrey from the Department of Medicine. But without any further ado, we hand you over to our chair for the afternoon, Graham Mankies.
Thanks very much, Mark, and welcome again to our UCT Department of Medicine COVID-19 ECHO webinar. We've got a full program this afternoon. We've changed the format slightly. Usually we start off with an epidemiological update, but Andrew Poole can only join us at the end of the webinar, and so we'll be giving a very brief Western Cape epi update towards the end of our webinar after we've had the main talk and the panel discussion. So the main focus of our webinar this afternoon is on the Coronation trial, which is an international multi-center trial looking at prevention of COVID-19 in healthcare workers. And we're going to have two talks. The first talk, it's a really great pleasure and honor to welcome Mihai and Netea as the first speaker who will provide the scientific context for the discussion of the Coronation trial. Mihai is the professor and head of division of experimental medicine at Radboud University and Nijmegen Medical Center in the Netherlands. And he's really internationally recognized for the work that he and his group have done in leading the field of what is termed trained immunity, which in simple terms is the innate immune system's version of immune memory. And that concept of trained immunity really provides the theoretical underpinning for the first intervention that's going to be assessed in the Coronation trial, which is the MMR vaccine. So Mihai, very grateful that you can join us this afternoon and provide us with an overview of trained immunity.
Thank you very much. Thank you for the invitation, and it's a pleasure to be together with you today and to be able to share some of our data. And I hope everybody can see my
slides
now. So I would like to discuss with you briefly in the next 10-15 minutes at most, a little bit about the concept of trained immunity, which is a concept of an innate immune memory, which is helping us to defend against severe infections. And I would like to start this, and actually most of my presentation will be focused on Bacille Calmette-Guerin BCG vaccine, which is used for almost 100 years, because this was one of the vaccines that was most studied in the context of heterologous effects against other types of infections than tuberculosis. But I would like to point out that the same scientific underpinnings are also true for the measles vaccine. For the measles-containing vaccines, such as MMR, there are studies ongoing now. We are also doing such an immunological study, and we believe that similar type of mechanisms are at play also for MMR. But just to give you the scientific basis of the concept, I will use today the BCG vaccine. And BCG vaccine has been observed to have heterologous effects very early after its introduction in various European countries. And this is one of the first observations made in Sweden in 1932, when introduction of BCG led to a very significant decrease in mortality in the young children vaccinated from 11% to 4%. This was, of course, before the antibiotic era, so a lot of children unfortunately died of infections, and that could not explain by TB deaths, because the TB deaths were actually a very small percentage from the total mortality. So it has so the doctor who introduced BCG, Carl Nestlund, concluded that there must be a sort of non-specific immunity induced by BCG. And this type of observation has been made many times that BCG was introduced in various countries, in United States, in Africa, in South America, and so on. And this is an overview that was written by Higgins and colleagues in British Medical Journal in 2016, and everything which is on the left of the line is decreased mortality in a BCG vaccinated group. And you can see here there were a lot of epidemiological studies showing that BCG administration decreases all-cause mortality. And there was observation also that it's especially respiratory tract infections and neonatal sepsis, which was decreased in these young infants by BCG vaccination. And there are also, in the meantime, some additional studies in adults and elderly. One paper in adolescents in South Africa, actually beautiful paper in New England Journal of Medicine, with revaccination with BCG for TB, but people have looked in the safety, as a safety precaution at the incidence of respiratory tract infections, and they observed that in the BCG vaccinated adolescents, there was 73% reduction of these infections. And there are two smaller studies, one in Indonesia and one in Japan, done in the elderly, showing also less respiratory tract infections if people were vaccinated with BCG. Now, the same type of observation has been made in regards with measles-containing vaccines. Its effects on all-cause mortality were summarized in the same BMG paper in 2016, and the people from the WHO SAGE committee who put together this report concluded that it's likely that measles-containing vaccines also have a beneficial effect on all-cause mortality. And this is basically one of the main reasons for following that up further in our case in the Crown Coronation trial. Just very briefly, how is this working? And we showed actually that in animal studies that BCG can protect against non-mycobacterial infections, in this case candidiasis, but other people have done it with other bacterial and viral infections. It has been done for influenza and so on, in a T and B cell independent fashion, because the same type of protection can be obtained in an experimental model in mice who are lacking an adaptive and functional adaptive immune response, such as SCID mice. These are the mice non-vaccinated with BCG that die from candidiasis. And if we vaccinate the mice with BCG 2 weeks previously, we see a complete protection of the mice. And we followed these experimental studies also with studies in humans, proof of principle studies in humans. And we have used as a model of experimental infection in human the yellow fever vaccine, because the yellow fever vaccine is an attenuated virus, and we can follow it by performing a PCR in the blood, and we can see the viremia for up to 7 days. And when we followed up yellow fever vaccine viremia in individuals who got placebo or BCG 1 month before, what we observed is that on day 3 and 7, very few people had viremia, which was normal, especially on day 7. We eliminate the virus. On day 3, not everybody showed virus yet in the blood, but on day 5, almost everybody had yellow fever vaccine virus in the blood. But we have seen a very significant decrease in this viremia if the individuals got BCG 1 month before. And that was not accompanied by a decrease in yellow fever antibody titers. Those were perfectly normal 3 months later. So we could conclude based on that there is a clear effect of BCG on a viral infection, in our case, induced by yellow fever vaccine, and it can protect against a mild heterologous infection in humans. And this protection was associated by an increase in the function of innate immune cells. We know that basically in all the immunology books, it's the lymphocytes, either B or T lymphocytes, which develop in memory cells after an infection, and that is meant to protect us very specifically against a pathogen. What we have observed, however, after BCG vaccination, is a very broad enhancement of the function, not of lymphocytes, of adaptive immune cells, but of monocytes and macrophages, cells of the innate immune response, which increase their production of pro-inflammatory cytokines after the BCG vaccination, independently whether we stimulate them with Mycobacterium tuberculosis, which is a specific infection against which BCG is meant to protect, but also when we stimulate these cells with Staphylococcus aureus or Candida albicans, so totally unrelated microorganisms. This function of the monocytes is actually exerted at the level of the myeloid cell progenitors, because monocytes are in the circulation only for a couple of days, so we cannot explain these long-term effects for months and even 1 year that we observed. The only possible explanation, and that is what we have shown, is that BCG vaccination induces very strong changes in the myeloid cell progenitor in the bone marrow. And this is a different study in which volunteers kindly agreed to donate not only blood, but also a bone marrow aspirate. And when we purify the hematopoietic stem cell progenitors, what we have observed is that the transcriptome of these hematopoietic stem cells is very different after the BCG vaccination, with a strong shift towards myelopoiesis and towards activation of myeloid cells. What is basically the in-depth mechanism of that? It is restructuring of the chromatin at the level of the DNA in innate immune cells and in the myeloid cell progenitors in the bone marrow. In naive cells, there is a very tightly packed chromatin in the nucleus of the myeloid cells, and those cells do not react basically, or can react very difficult upon stimuli. They don't produce proteins and so on. If we activate the myeloid cells during an infection, there are chemical processes that change the chromatin structure, not necessarily structure, but change the chromatin architecture. There is methylation and acetylation of the histones in the nucleosomes in the nucleus, and that leads to opening of the chromatin. So transcription factors can bind much easily to chromatin. They can initiate gene transcription. And we always thought it's like opening a book. It's if in a naive, in a non-stimulated cells, the DNA is closed, the chromatin is closed like a closed book, you cannot read it. When an infection takes place, you open the book to read the instructions, how to make the proteins that are necessary for host defense. What we always thought is that after the infection, all these chemical chain modifications are lost, and basically the book is closed and the transcription stops. And that happens indeed, but there are some chemical modifications, especially methylation at certain places of the histones, which mark the proper genes that are necessary for host defense in these cells. It's like putting a bookmark in the book. So the next time that we need to read the instructions how to make the the proteins necessary for host defense, we can much easier open the book at the right place and produce much easier these host defense proteins. So this is exactly what we think is happening. And this is associated with an increased immune response, enhanced innate immune response, the second time that the organisms encounters an infection. And what is also very important, this second infection does not need to be the same as the first one. It is not a very specific process. This is an overall process through which the first infection boosts the innate immune mechanisms, and the second time we encounter an infection, that the response of the innate immune cells is better, it is improved. So how do we think such processes can help us in case of COVID-19? Well, if we think of non-vaccinated individuals with low innate immune responses, then can lead to easy multiplication of the virus with high number of virus particles in the circulation, high viremia, and that can lead to hyperinflammation systemically, a lot of symptoms, high severity, and unfortunately in some individual death. We want, with MMR and with BCG and with these non-specific attenuated vaccines, to increase the innate immune responses So the initial multiplication of the virus is much lower. It cannot multiply easily. That can lead to low inflammation, low symptoms, and survival. And to show you that, that this has a good chance to be successful, these are the results of a study that we published very recently, 2 weeks ago, in which 200 elderly were randomized to receive either placebo or BCG vaccination. And this was done before the COVID-19. And we followed them up for 1 year to see who's getting the infection, whether the BCG vaccinated elderly get more or less infections. And as you can observe here, there were 40% less infections in the BCG vaccinated group. And this is a total number of infections. But when we split up in the type of infections that were prevented by BCG vaccination, actually we see no effect on intra-abdominal infections or urinary tract infections and so on, but we see very strong impact on respiratory tract infections. The individuals, the elderly who got the BCG vaccinations had 80% less respiratory tract infections than those with placebo. And that was in line with the previous studies showing the same thing in children and adolescents. Finally, what the concept behind the entire Crown Coronation trial with MMR and also the BCG studies that are ongoing around the world is that during the emergence of a new pathogens and before we have the chance to develop and use a specific vaccine, there is a period of 1, 2, maybe 3 years sometimes, and this period can be covered by the use of attenuated vaccines, which can induce strain immunity and a non-specific protection against infection. And this is exactly what we would like to propose in the Crown Coronation trial. And finally, of course, these are all the colleagues of us who are contributing to these studies. Thank you very much.
Thanks very much, Mihai. That's a really authoritative and accessible summary of that field of trained immunity, and really sets the stage for our discussion of the Crown Coronation trial. I'm not going to take questions or discussion at this stage. We will take it after the second talk and our panel discussion. I just also wanted to say to people that attending the webinar that Mihai and a colleague have recently published a piece in the New England Journal of Medicine that summarizes the issues around trained immunity. I think it was published last week. Is that correct, Mihai? Yeah. So that's another source of information around this issue. To move on to our second talk, it's going to be a summary of the Crown Coronation trial from Bruce Biccard and Sinead Delaney-Moretlwe. They are the national co-PIs of the Crown Coronation platform trial that is looking at interventions to prevent symptomatic COVID-19 in healthcare workers. Bruce is the second chair in the Department of Anesthesia and Perioperative Medicine at Groote Schuur Hospital in UCT. And Sinead is a research professor and director of research in the Wits Reproductive Health and HIV Institute at the University of Witwatersrand in Johannesburg. And they will be discussing the clinical aspects and plans with respect to the Crown Coronation trial. So over to you, Bruce and Sinead.
Thanks, Graham. And thanks to all the colleagues and affording Sinead and I this opportunity. And also Mihai for really creating the foundation for this trial. So we're going to quickly just run through what the Crown Coronation trial is. And our first intervention is MMR vaccine. So basically, we're all well aware that SARS-CoV-2 has been a massive public health problem, particularly with no prior immunity, rapid transmission, and a spectrum of disease of which about 20% can be severe. And to date, there's over now 29 million cases and nearly a million deaths. And it's caused massive social and economic disruption, and particularly, as we're well aware, in South Africa. And we still have few proven interventions. And so we need robust data and we need good clinical trials to identify potential interventions. The primary objective of the Crown Coronation is to determine effectiveness of select interventions in preventing symptomatic lab-confirmed COVID-19 in healthcare workers. And why are healthcare workers? Well, it's a vulnerable population. They have repeated exposures, often to large inoculums. They need to travel frequently for work, and PPE, unfortunately, is not always available, and it's not infallible. And we've seen in low resource environments that epidemics can have a devastating effect on the healthcare system. For example, Ebola, where in Liberia, I think they lost about 10% of their healthcare workers. And what it results in is also increased all-cause mortality unrelated to the epidemic itself. So the Crown Coronation trial is a Bayesian platform adaptive trial. And so this is a very efficient way of identifying potential interventions with efficacy. So the Bayesian principle, meaning we use data from interim prior analyses, which we can use to make informed decisions on whether we should declare intervention futile or potentially superior. The platform nature, in that we've got a master protocol on which we can add interventions over time, and the adaptive nature allows you to add and remove interventions as you go. So at the moment, we are starting with MMR with approximately 5,000 participants and are hoping to add a second arm after that. And so at that stage, it would become like a 2x2 factorial trial. And then hopefully after that, we could even add a third arm. The trial will be run in a number of countries. It's already recruiting in the States. Also it will also be run in the UK, Ireland, Canada, South Africa, and then into Africa as well. The outcomes, primary outcomes, symptomatic, lab-confirmed COVID-19 after 60 days after the intervention, and secondary outcome is a WHO severity scale for COVID-19. The sample size for MMR for this first stage is 5,000 patients, and that's it's got sufficient power for event rate at 5%, and of which we hope to contribute 2,500 patients or participants, at least from South Africa. It's 1-to-1 randomization, it's block randomization and stratified by age and site. And then obviously through the Bayesian approach, we can add or remove arms as necessary. I'll hand over to Sinead now, thank you.
Thanks, Bruce. So I think sort of the key question is, what is the evidence that we have that vaccination with MMR could prevent COVID-19? And Mihai has laid out a very elegant explanation of trained immunity. But what do we have in the way of evidence to support our selection of MMR? And there are probably three key points. The first is that some ecological data supports the idea that countries that have extensive MMR vaccination coverage appear to have lower COVID-19 severity. And it's also been speculated that perhaps recent vaccination status is why we're seeing less severe disease in children. There is also observational evidence that was first generated, obviously, the data that Mihai shared showing that live attenuated vaccines are associated with reduced all-cause mortality. And then we started to see observations emerging suggesting that perhaps MMR is associated with less severe disease. One example is a cohort of naval recruits who are immunized with MMR prior to entry into their training, and out of almost 1,000 young recruits who had SARS-CoV-2, only one was hospitalized for severe disease. But perhaps stronger data has come from an analysis of about 140,000 individuals who are part of the Mayo Clinic electronic health records. And an analysis was done of those records between February and July of this year, really looking at SARS-CoV-2 test outcome status and history of vaccination in the past 5 years. And in that analysis, individuals that had been vaccinated with MMR had an almost 50% reduction in SARS-CoV-2. So a significant amount of observational data starting to suggest that this could support that hypothesis. And then you've heard the explanation from Mihai that this may be underpinned by this concept of trained immunity. And then next slide. But also that there may be an element of adaptive immunity and cross-protection between the antibodies generated by the MMR vaccine, either from rubella or measles, that have sequence homology with the spike glycoprotein, which may also provide additional protection. Next slide. So there has been increasing interest this year with the idea that live attenuated vaccines may provide some benefit against COVID-19. And there were a couple of articles in The Lancet and Science really articulating the potential value of either BCG or an article on oral polio vaccine. And this is really translated into a significant number of trials when you look on clinicaltrials.gov. So about 18 trials evaluating BCG for prevention of SARS-CoV-2 and associated COVID disease, involving about 40,000 participants in many high-income countries, but also in a number of low and middle-income countries. And obviously, the TASK trial in South Africa is evaluating this concept. There are 2 trials that have been launched looking at the oral polio vaccine, and those are involve about 7,000 participants. And then with MMR, apart from the CROWN trial, there are 2 small trials that have been launched in Egypt and the United States. So a significant interest in the potential of live attenuated vaccines to prevent COVID-19. Next slide. So I think one of the key questions that we get is why a non-specific vaccine? Why wouldn't a healthcare worker want to participate in a SARS-CoV-2 specific vaccine trial? And as the consortium, I think we strongly support the development of specific vaccines, but recognize that there's a long path to the development of proven vaccines, and there may be many stops along the way. And by comparison, what we have is an intervention that's well known, likely to be inexpensive. I think MMR costs about 250 Rand in South Africa. It's a simple intervention. It's once off. We have a very well-known safety database, and this intervention is well tolerated. Also, there are very few contraindications. And for that reason, this makes it a really scalable intervention in the short term while we wait for specific vaccines. Next slide, Bruce. But I think what's also interesting and well worth testing is the idea that the non-specific vaccines could be combined with specific vaccines to potentially enhance their response in groups like the elderly who may not have good responses to vaccines. And because of the non-specific approach, there's also the idea that MMR may be useful not just for this pandemic, but potentially for future pandemics. And for this reason, we think that these questions are important to address. So who can join the CROWN Coronation trial? Well, it's targeted at healthcare workers who are able and willing to give consent and of legal age to do so. And participants can enroll if they have no contraindications to MMR, which is essentially no evidence of severe immunosuppression. And so what that means, in fact, is that we can enroll people living with HIV as long as they are asymptomatic and generally on treatment and well controlled. So one of the novel aspects of this trial is that it is a light touch remote trial. We can register participants through the website, and the link is there, and you're welcome to go and have a look. participants are essentially assessed for eligibility through the website. If they're eligible, they are randomized. They're enrolled onto a weekly SMS system, which is used to assess symptoms or adverse events. And they also are required to answer a monthly questionnaire. If they become symptomatic, at the time of vaccination, they're given a kit with swabs where they can collect nasal swabs, self-swab, and those can be collected or picked up from drop-off points. Also, we collect DBS at baseline and at several time points in order to understand potential exposure and asymptomatic infection. And the primary outcomes are going to be assessed at day 60, but we follow up through to day 150. So we're very excited about the Coronation trial. We think that it will generate some really important information. It's going to teach us, I think, about a number of things, but also provide some really practical benefits for participants, including prevention of measles, and this is a concern given sort of declines in measles coverage or immunization programs. There is also the potential to protect against COVID. And in future, we may be able to answer the question about generating improved responses to SARS-CoV-2, as well as to infections in general, making this a very useful intervention in the future, as well as providing, I think, important information about the choices and preferences of adults in the field of adult vaccination. So we hope to launch in South Africa in the coming weeks, pending SAPRA approval, and look forward to taking your questions. Last slide. But before we take questions and begin the discussion, just to acknowledge the COVID Therapeutics Accelerator, which is the main funder of this trial, as well as the South African Medical Research Council, which is supporting the South African sites, and the many partners involved in this trial. I think many of us involved in large clinical trials know that it takes a village to launch and raise a trial. So many thanks to all of those who've been involved. Happy to take questions.
Great. Thanks very much, Sinead, and thanks, Bruce, for that very clear overview of the trial. We're going to move on next to the panel discussion, and I just want to introduce the three panelists. The first is Linda Gale Becker from the Desmond Tutu HIV Centre and Department of Medicine at UCT. Then Netea-Moretlwe. Netea is from the Department of Pediatrics at UCT and Red Cross Children's Hospital, as well as an expert in vaccination, working with Vaccines for Africa. And then Johnny Peter, who's from the Division of Allergy and Clinical Immunology at UCT and Groote Hospital. So I want to ask Netea first if he just wants to speak to the issue of the non-specific effects of BCG from the perspective of a pediatrician, and then if there are any questions that he wants to address to the panel regarding the Coronation trial or some of the vaccination issues in general.
Thanks, Graham. I hope I'm unmuted now and you can hear me.
Yeah, that's great. Yeah.
Okay, thank you. Thank you very much for a very interesting talk. And one thing that I think is very clear from pediatrics point of view is that a lot of this phenomenon had already been observed. And I think a lot of the work of Peter Abbey in Guinea-Bissau showed this impact of BCG in terms of reducing all-cause mortality in children. So in pediatrics, that is, as Mihai pointed out from the beginning, is a phenomenon that was observed right from the outset of the use of BCG, of its impact on all-cause mortality in children. What is interesting, though, with BCG is that not all BCGs are the same. So, for example, BCG, the Danish strain, for example, has got a much different impact than, for example, the BCG Russia strain. In fact, the effects that we're talking about in BCG have been very minimal when you look at the Russian strain. So it's not, BCG is not just BCG. And I think that's what we've been worrying about even in this country when we ran out of the Danish strain a few years ago, that there's some anecdotal evidence that there may have been an increase in TB, for example, but also the non-specific, those heterologous effects of BCG don't seem to be the same from different strains. Also, what is very interesting, apart from even with the measles use, again, which Peter Abi showed in Guinea-Bissau, is that having another non-live vaccine after that seems somehow to impact on the positive effect of the live vaccine. The live vaccine has to be the last vaccine given for you to benefit from that major impact on all-cause mortality. And that's why even when I was reading this about this trial, I wondered what happens when you encourage healthcare workers, for example, to get immunized against influenza or other things where you use a non-live vaccine. Is there any chance that you may blunt the effects that the live vaccine actually gives in terms of protection against other respiratory pathogens? Because in children, if you look at them, even in observational studies, the effect disappears. In fact, the same systematic review from Julian Higgins that Mihai was talking about shows an increase in mortality due to DTP, to non-live vaccines in the very same systematic review. So the question here is, what then becomes the impact of giving the measles vaccine or MMR vaccine for that matter, and then having a non-live vaccine which you use down the line because you still want to protect healthcare workers, let's say against pertussis or against seasonal influenza? Would that blunt the effect at all, and then you lose the impact of that vaccine? So that's just a few things that I would just like to comment on. Thank you.
Thanks, Rizan. Mihai, any thoughts on those comments?
Yes, I think you are perfectly right in all the points that you raise. And the BCG strain is very important to start with the beginning. It's very important indeed. It has been reported that probably BCG Denmark and BCG Japan are more, let's say, have more boosting capacity than BCG Russia. That is true. In our hands, it is also true, although we do see also some effects also with BCG Russia. It is a question at a certain moment, hopefully, that people would recognize these differences between the various strains and try indeed to improve the availability of BCG Denmark and Japan. But I do think that even BCG Russia has some effects. Regarding the order of vaccination, this is also a very interesting and very important work that has been done by Peter Abi. And indeed, he observed in children that this is the case, that at least partially there is a negativation of the effects of a live attenuated vaccine if you give DTP, for example, at the end. This is something that needs to be studied in adults. That has never been done, as far as I know, in adults. I do see a couple of small indications in the literature, and we have the same observation in our hospital, where we used the live inactivated influenza vaccine last year. It seems, however, that for COVID-19, so I'm not talking in general, but specifically for COVID-19, even the influenza vaccine, inactivated influenza vaccine, has a slight beneficial effect. So, and that is also coming, I think, from the database of the colleagues at the Mayo Clinic, showing also that that influenza had such a positive effect. So I do think that for influenza, which probably will be given also in the hospitals together or in addition or so on with MMR, in our case with BCG, there will still be some positive effects against COVID-19.
Thanks. So then to move on, before coming to Linda Gale, I wanted to ask Johnny Peter if there's some comments that he wanted to make, and specifically to mention that Johnny is an investigator on one of the BCG trials that's looking at BCG for prevention of COVID-19. So Johnny, do you want to come in and make a few comments, and if there are any questions that you have to any of the speakers?
Yes, thanks, Graham. Yeah, so, you know, we, or at least I find the idea of trained immunity a very attractive sort of hypothesis. And I do think, though, that there are several factors that may kind of influence the efficacy. And I think Ritwani, mentioned this issue about the timing of vaccines. And I think specifically in relationship to BCG in the South African context, the issues that we felt are relevant and part of the justification that we had to actually have a BCG trial in South Africa is really the issue of, prior BCG vaccination, number one. Number two, our very high burden of TB, and also the high burden of non-tuberculous mycobacterium. So I'd like to, get Mihai's comments on how those kinds of, things might mitigate the effects of BCG. To give some context to how we're doing with our TASK coronation study, or coronavirus study, sorry, the coronation study is a different study. But how we're doing is that it's sponsored by TASK, and that's led by Andreas Dekon. And we've each committed to do 500 participants. And so we're now nearing the end of enrollment. We're up just over 850 participants at the moment. So our target for that study is 1,000. Our primary endpoint is actually looking at hospitalization due to COVID-19, and this has been lower than expected. And so we're looking probably soon to amend the trial to target both populations that are excluded from the first phase of COVID-specific vaccine trials. So potentially looking at the elderly, and you saw Mihai's data on the Activate trial, and then also looking at potentially people with morbid obesity or other groups that would be excluded from conventional COVID vaccine trials. Because, and I'll be interested in the Coronation investigators' comments on this, but our personal experience so far has been that since the availability of COVID-specific vaccine trials in Cape Town, we found it harder to recruit for this BCG trial, and I think that's a potential factor that will also affect Crown Coronation. And so I think that we're almost done, and we probably will broaden it out to improve our study power. But that's the current situation for the BCG trial in South Africa. I think it's been globally an interesting thing. So although all those trials were listed, the 18 trials and huge number of participants, actually the drop-off in COVID numbers and COVID cases, I think will mean that several of those trials will have to run longer to potentially get to the adequate power to generate results. So I've been interested in the fact that we haven't yet actually been able to provide even a first result from one of these bigger trials on BCG. So I think that's also another challenge that we'll have to face in terms of investigation of COVID in general, and I think it applies to COVID-specific vaccines as well. So I think in terms of my questions, I wouldn't mind. I'd love to hear Mihai's comments on how he feels things like non-TB mycobacterium might affect. High levels of environmental mycobacterium might affect the non-specific effects of BCG. And then in relationship to this, and I think this pertains to both the MMR and the BCG from a basic science perspective, is I think it's quite interesting what the actual molecules are in the particular live vaccines that are having the effects. In terms of toll-like receptor agonists, or one of those groups that might be at play. Because the reason I say that is I also think that there might be other factors environmentally. I know Tobis Coleman has got some lovely data looking at toll-like receptor responses that are affected by other environmental factors and could be more fluid in a way. And I think that might affect the sort of longevity of the effect of non-specific protection as well. And then I guess from the Crown Coronation trial panelists, I'd like to ask the question around, do we think, so your primary endpoint in the trial is looking at infection, so COVID infection. And I think it's quite interesting, like to think of a non-specific effective vaccine affecting actual infection as opposed to sort of mitigating severity or, in terms of decreasing viral loads. So maybe you could make some comments about that. I'll leave it there. Thanks, Graham.
Thanks, Johnny. So Mihai, the initial questions regarding environmental mycobacteria and the influence that they might have?
So regarding that, actually, there are no direct data about that, but I'm pretty optimistic that a high environmental mycobacterial load would not influence too much, I think, the non-specific effects for two reasons. One is the epidemiological reason, because we see these effects happening. I mean, for example, the study of Elisa Nemes in adolescents with revaccination was in South Africa, basically, and it was showing very clear non-specific effects, although they were not looking for the non-specific effects, but they were still observing them very clearly. And the same is all the studies in West Africa, also the same situation of Peter Abbey showing these effects. So that is the, let's say, the epidemiological argument. And there is also the immunological argument based on the way in which, let's say, an environmental mycobacteria would act at the level of the mucosa, which is very different immunologically from what is happening with BCG being given intradermally, where the immunological mechanism is very different from ingesting something or inhaling something. And because the mucosa are meant to be tolerant, but when we give things intradermally, we stimulate the immune system. So yeah, I'm relatively optimistic, let's say, regarding that.
Thanks, Mihai. So Bruce and Sinead, just Johnny's questions about challenges with respect to enrollment, and then secondly, the issue of the endpoint.
Yeah, maybe I can start with the straightforward one about the outcome. The outcome is symptomatic laboratory confirmed COVID-19. So it is a disease outcome focused on symptoms, but that is laboratory confirmed, so that we're not confused by any other symptoms. And there is a secondary outcome that is focused on severity, using the WHO scale to make some kind of assessments of whether we can mitigate severe disease. The reason to test serology is to some extent to understand what sort of prior exposure there has been, and so to potentially exclude those people in a per protocol analysis, so that we can maybe understand the effects in the population that wouldn't have been exposed. But the outcomes are disease outcomes. I think for the reasons that you're outlining, which is that what we really want to do is keep healthcare workers from not getting ill for prolonged periods of time. And that's sort of the primary goal. Bruce, I don't know whether you want to take the recruitment comment.
Yeah, that's fine. Thanks, Johnny. Obviously, this is difficult, although I believe there's a large number of healthcare workers, and we've got a broad platform of predominantly frontline workers, you know, both in the hospitals and in the community. And we've built up a large collaborative across South Africa and also in other countries. And I think with that sort of collaboration, we should be able to get to the numbers. We believe we can get to the numbers that are needed, but I agree it's difficult, and it's difficult because we've gone over the hump. And so, you know, I suppose the anxiety is maybe there's less anxiety now with some healthcare workers, so maybe recruitment might be a bit more difficult, but we got a broad platform to work from. And also, I think the same as with your BCG trial, you know, having the opportunity to participate in a non-specific vaccine with a long history, a long safe history, is very desirable to a healthcare worker. And so let's hope we can pull it off, because it's certainly important. Even if we miss this pandemic, a coronavirus pandemic is definitely going to happen again.
Can I say something, Graham, as well, that maybe others can comment?
Sure.
I mean, I think one of the things that is interesting, the South African Immunology Society had Christina Ben talking as well about BCG. And I think one of the aspects that is a challenge for COVID-specific vaccines is, of course, that the virus might change. It's taken us a long time to get a vaccine for influenza. We still don't have a universal flu vaccine. And so yet it's quite undefined. And I think that, you can make an argument. So the one argument, which I think is a valid argument, which is the safety history of vaccines like BCG and MMR. But the other argument is a broader, a broader brush, where actually a COVID-specific vaccine should the virus mutate sufficiently to have a vaccine like that not be specific, then you actually might have a situation where a broader non-specific effect might be more beneficial. So I think that's another factor that I think is worth considering.
Yeah, thanks, Tony. And I mean, as Sinead went through her slides, I mean, one of the arguments for this is really that there's absolutely no guarantee that we will get a successful SARS-CoV-2 specific vaccine. I mean, people are optimistic, but we know that there might be impediments in the way to discovering that. So, that's why we need to look broadly, and studies like this are important. I just want to bring in Linda Gale, and Linda Gale Becker from the Desmond Tutu HIV Centre is one of the investigators on the Crown Coronation trial. And I just wondered whether you wanted to expand on some aspects or address any questions to the panel, Linda Gale.
Thanks, Graham. Yeah, thanks for this opportunity as well. We appreciate the prime time. I think I do want to direct a question to the colleagues on the Crown Coronation leadership, and I think it's an important one. It is possible also, and I'm an avid vaccine fan, but that initially we may not see as good, vaccine effects in HIV, those people living with HIV, and of course healthcare workers living with HIV are important for us. So Sinead, you alluded to the eligibility criteria, but do you want to just sort of speak a little bit more about how we hope to involve healthcare workers living with HIV, and obviously this may be somewhat different in the BCG trial. And then I do see a question here to Mihai. It's a more difficult one, but also, passionate about this, just trying to understand the BCG, and obviously ELISA's study, we excluded latent TB infection in that study. So what is the role, in Africa, where we obviously have Mycobacterium tuberculosis almost vaccinating in its own right? So I just wanted you to sort of expand on that a little bit, and the durability of trained immunity, these myeloid cells. But back to Sinead and Bruce in terms of our healthcare workers living with HIV.
Yeah, thanks, Linda Gale, for that question. I think we think it's really important to include people living with HIV in these trials. I think we've observed, and it's data that came from the Western Cape, suggesting that people living with HIV are at higher risk for severe disease, even though it may not be as high risk as some other comorbidities. And so I think it's important that we generate data around sort of the safety and efficacy of these interventions in people living with HIV, particularly in a place like South Africa where HIV prevalence is so high. I think the useful or the important things to know about MMR are that it has been generally found to be safe in people who are not severely immunosuppressed. The Department of Health Vaccinators manual suggests that people living with HIV who are asymptomatic be vaccinated, as do a number of sort of guidance documents, and essentially anyone with a CD4 count above 200 but living with HIV could receive an MMR. There was an interesting WHO systematic review done in about 2009 that looked at the safety of MMR in people with HIV who had been vaccinated, and essentially that was mainly in children, but in about 5,000 children vaccinated, there was only one adverse event associated with immunization, and it was not specific. To some extent, this was a little bit confounded with HIV disease, so I think it's still important to investigate this further, and I think being able to do the trial in South Africa will create the opportunity for us to demonstrate safety in people who are not immunosuppressed. To do this, we will obviously encourage people to test. And so I think there's secondary benefits, as with many trials, in terms of encouraging healthcare workers also to know their status.
Thanks.
Great. Thanks, Linda Gale. So I'm going to just move on to seeing if there are any questions from the chat room to bring in Mark again.
Okay, so there are a few questions. I think the first one may be addressed to Razani. Does the immunization schedule in children explain why they are less likely to acquire infection? So maybe Razani, you can address that one.
Is that specifically with respect to COVID?
In terms of the
less likely to get COVID.
Immunization schedule, does it mean because they're getting it earlier, they're less likely to get COVID or long term?
It's been very difficult to actually find out the reason why. I mean, there have been a number of hypotheses, including the ACE receptor, how much they exhibit of the ACE receptors, and if there's a dose, age-dependent dose, so to say, in the increase of expression of ACE receptors that are required for COVID, and that may partly be related to their less susceptibility for infection, because what we're talking about now with other vaccines is tends to speak to live vaccines that have got the impact of immune training that we're speaking about. And the current South African schedule after BCG really follows the whole way forward with 9 live vaccines that one would have expected them to dampen any kind of protective immunity that would have been generated by those live vaccines. If Peter Abbey's observational studies have anything to go by, I think it may be something else different from the vaccines themselves. But I'm not really sure.
Mihai, any comments on that in terms of the timing of the schedule, in terms of long-term protection?
Sorry, I'm not so sure to whom the...
So just in terms of, I mean, we get the BCG and the, spread after birth, we have the early immunization. Does that actually protect us, or do we need boosters later on in life?
Well, I think that at least on the immunological level, and I think also epidemiologically, we do need later in life a second vaccination if we want to be protected, because I think that the non-specific effects will be probably a couple of years, maybe I would guess at most 3 to 5 years, honestly. So I think that for later protection, we do need to get a new vaccination. With regard to the questions that were also in the chat and also asked previously regarding the latent infection with TB, there is one study actually showing that the latent TB is also associated with improved function of innate immune cells. There was only one study done by Tom Otterhoff in Leiden, I think 2 or 3 years ago. That being said, again, I do think that there is an amplification by BCG vaccination, and we are actually busy now with a study in which we even try to give more than 1 BCG doses to see what is happening if we try to amplify the effects.
So then there's another question relating to the flu vaccine. Would that not be better than the MMR in view of the drive to vaccinate against flu to prevent severe influenza and pneumonia during this COVID? So should we not be pushing the influenza vaccine rather than the MMR? Maybe Bruce or Sinead?
So I think that's a great question. I think sort of the reason to select MMR is that it's one of the live attenuated vaccines, and these effects have been thought to be or have observed most strongly with live attenuated vaccines. But certainly the recent Mayo paper identified a number of other vaccines where we may see these effects, and I think that this is where the Coronation trial can come into its own, in the sense that we can if we want to add additional vaccines and look at the effects of them independently compared to placebo, but also look at them in combination to see if you get additive or synergistic effects. So those are questions still to be answered.
Bruce, any comments from your side?
No further comments. I think Sinead summarized it well.
Wendy, can I just ask Mihai, does does the same experimental data exist for the influenza vaccines that you've showed, or?
Not that I know. There is an ongoing study, I think, in Stanford that was started before the COVID-19. And as far as I know, it is not yet published, but I have seen it presented last year at a conference, showing some data also with influenza vaccine. And we do have some very preliminary data in vitro that influenza vaccine can have similar type of effects as attenuated vaccines, but the data are much less solid and much less broad than what is known about BCG.
And Graham, can I ask Mihai as well if there's been any head-to-head comparisons of these live attenuated vaccines in vitro even?
No, there were no such comparisons. We are actually comparing now. There is an ongoing study in our group now in vivo in healthy volunteers looking and comparing BCG with MMR and the combination of BCG plus MMR. But we'll know that a little bit later towards the end of the year.
Great.
So one last question before we go on to Andrew. With regards to the question of environmental microbacteria potentially affecting trained immunity or boosting the effect of the BCG vaccinations, how might this be affected by the phenomenon whereby environmental microbacteria levels affect the efficacy of BCG vaccination? So that's open to anyone who would like to go for that one.
As far as I know, nothing is known about this, and I think it's a very good question. What we would need actually is to compare the response to BCG vaccination in environments where there are less or more environmental mycobacteria. In terms of the non-specific effects, I don't think that these studies have been done yet. I would not be surprised to be an effect. I do think, though, that in both situations with high or low environmental mycobacteria, there will be effects of BCG.
Address most of the questions.
Okay, great. So we're going to move on for the last talk of our webinar to a brief overview of an epidemiological update of the epidemic in the Western Cape from Andrew Bull. So Andrew, I believe you're online.
Yes, I'm here. Can I share my screen or?
Andrew, I'm putting your slides up now.
You're going to project, okay.
Now, there we go. We should be ready to go. Yes, please proceed.
Yeah, okay. Next slide.
My apologies, Andrew. It's just jumping out all the time.
Okay, so it's going to very brief this week. So just to give you an update on the trajectory for the different metrics that we've generally been presenting. It's continued trend as we've seen in previous weeks. The one change that I'll point to is that the hospitalizations hadn't been declining as fast as the other metrics, and what we found is that quite a few patients who were diagnosed with COVID quite a long time ago, now that we've been at this for some months, were being admitted subsequently, and that was registering as COVID admissions. And so we've put a limit of 21 days after diagnosis in order to be counted as a COVID admission, and that's brought those extra admissions accounted for about a third. So you'll see the blue lines coming down a bit more precipitously than they had in what we've shown previously. But otherwise, the week-on-week picture is very similar to what we've seen over the last few weeks. Next slide. What has changed this week is that the Medical Research Council, who release the excess deaths by province and by metro every week, have changed their baseline for the Western Cape. So you'll see that on the left-hand graphs, the top black line has reached the orange line, which is the prediction of natural deaths based on previous years. And we're still enumerating 100 or more confirmed COVID deaths every week in the province, so they would have had excess deaths lower, fewer than COVID deaths. So they've adjusted their baseline downwards, and they've provided a note in the report, if you look at it this week, and it adds about, on the numbers up until the 8th of September, it adds an extra 678 excess deaths to the Western Cape tally, which now sits closer to 6,000. And it also makes a tiny impact on the national, proportionately on the national estimate, which is now running at 99% of expected. Next slide. In terms of where there are, there's still ongoing high case numbers or clinical events or mortality, it's a pretty distributed picture with low numbers from lots of places. If there is one place that still has higher numbers, it's Garden Route, where Oudtshoorn in particular, for its size, has been a bit of a hotspot. Oudtshoorn has the highest age standardized mortality outside of the metro in terms of what we've enumerated. But the numbers are still low, and a few other places like Eastern, Klipfontein, Southern are still enumerating about 10 COVID deaths a week. But as I say, the numbers are small and distributed across the whole platform. Next slide. And that's it, just with the usual thanks to the various teams that contribute to maintaining these data. Thank you very much.
Great. Thanks, Andrew, for the update. So just to say thank you once again to Mihai, to Bruce, to Sinead, to all the panelists, to Andrew. We've had a really interesting webinar this afternoon, and to really wish the investigators in the Crown Coronation trial that best of luck, and we hope that they recruit rapidly and get a definitive answer to this fascinating question, which could potentially have a major role in both prevention of COVID-19, but also, as Sinead alluded to, potentially other pandemics in the future. So thank you very much, everybody. We won't be having a webinar for the next 2 weeks. We're going to be taking a break for the next 2 Wednesdays, and we will be back with our next webinar the first Wednesday of October. Back to you, Mark and Wendy.
Thank you, Graham, and thank you to everyone. And as Graham just indicated, that we'll be back in 2 weeks' time. And be well. And as usual, we want to continue saying hello to Prof. Saunders. We know you, Prof, and we trust you are well. Be well, everyone.